Our first speaker is Ken Johnson from Department of Plant Pathology at Oregon State University, and he will be talking about system approach to fire blight control in organic pear and apple without antibiotics.
Okay, welcome. My name's Ken Johnson. I'm at Oregon State University in Corvallis, Botany and Plant Pathology. We started a project a couple years ago now looking at fire blight control with antibiotics, and now we have an OREI project on this with the people you see here, myself and Todd, my technician, Rachel Elkins at UC Extension in Lakeport, and then Tim Smith down the road here in Wenatchee. And the rationale for the project is that antibiotics are prohibited under the eOrganic standard, and they've just signed this equivalency agreement. And our National Organic Standards Board just recently made a decision to sunset streptomycin and oxytetracycline kind of off the normal sunset schedule, and they're going to do it in after the 2004 season. And so that has us kind of scrambling about what to do about fire blight control if this comes about. The industry is still actively petitioning these against these decisions, but now they have to undo a decision, which is always a hard thing to do. And so my focus here is on floral infection in fire blight susceptible cultivars, which are the majority of organic pome fruit orchards for many of the reasons we've seen in the previous talks. They're the apples that sell, Galas, Fujis, Goldens, Honeycrisp, etc. And for what we're talking about here, we work in the West, so the strategies and the data shown are most applicable kind of to the semi-arid production systems that you see outside here. And what I mean by that is that we're not really trying to work this in around an apple scab control program. In apple scab, if you're really intensely fighting apple scab, then some of the things we're talking about here with biological strategies probably wouldn't work so well because of the number of sulfur and lime sulfur applications that have to go on to fight scab. So this is an important slide here just so that everybody gets this. So this is sort of an abbreviated disease cycle of fire blight. So the pathogen overwinters in old branches. These are called holdover cankers, and it actually is just along the edges of these old cankers. And in the springtime, it makes the jump from an old canker into the flowers. And then that's often unpredictable. It's very hard to predict. A lot of things can make this happen. Rain can make it happen. An ant can make it happen. A fly can make it happen. And so whether it happens or not is sort of a sporadic and unpredictable thing, but it does happen. Then the bacteria end up in the flower, and they grow on the flower, what we call epiphytically, which means on the surface of the flower, not causing disease at this point. And they can get pretty big numbers in a flower, 1 million to 10 million cells of bacteria in a flower. And then a bee comes along and pollinates the flower and moves to the next flower, often taking the bacteria with it. And so you get this little cycle we call the floral epiphytic phase, in that blows up the number of bacteria that are in the orchard. And then this, of course, this kind of thing, bee activity, bacterial growth, is driven by temperature. So the higher the temperature goes, the faster this goes. And that's really what makes the fire blight risk really climb in the springtime. So warm, sunny days in the springtime are when fire blight risk is going up. And then you get infection in the flower after you get a sufficient number in there, usually accompanied by a little wetting event, dew or a light rainfall can do it. It gets you infection, and then you get ooze, and then you get all the secondary phases that go on in the summer and making it a real headache for you if you're a pome fruit grower trying to fight fire blight and also grow a crop at the same time. So that's kind of the abbreviated disease cycle right there. So we had 4 questions that we were trying to answer in this project, and we want to know more about this activity thing here, about when does fire blight pathogen become active in bloom. And we've always had kind of sketchy data on this. Can we develop tools that would give us a better idea of whether or not the pathogen's actually out there or not? And then we were going back to some of the things like we're talked about this morning with apple scab, looking at what is the effect of a sanitation treatment. And here we're looking at a delayed dormant copper as a treatment, as a sanitation treatment on pathogen activity. We've been asking the question about bloom thinning, especially with lime sulfur. and how does that affect fire blight control? And that's been pretty interesting, and it does have an impact, and I'll show you a little bit of data on that. And then integrating products that we have that aren't antibiotics, can we achieve effective control of fire blight? So just kind of quickly here, I want to talk about this monitoring pathogen activity for a second. And we got into this technique, and it's still kind of a lab-oriented technique, but if you ask the question, is the fire blight pathogen in this bag of flowers? Like you went out and you sampled flowers in an orchard, and is the pathogen in there? And we've been using a little molecular assay that's a little like PCR, but it's simpler to perform, and you can run it in a heat block. It doesn't need a thermocycle or anything, and so and you can get an answer whether or not the pathogen's in there in about an hour. So you could do this with a petri dish, of course, or something like that, and plate it, and then 4 days later you might know if the pathogen's in there. But we can take a little bit of water out of this wash of flowers and run this DNA detection assay on it, and within an hour we can know if we can scout for the pathogen and whether or not it's in there. So we've been interested in that. We've been using this as a research tool, and so we've been doing things like this. I try to make this as simple as possible. We walk an orchard just like we're an IPM scout, pick flowers, and then run this what we call lamp scouting or lamp assay on each of these bags of flowers, and we can tell whether or not the pathogen's out there or not. And the interesting thing about this in the future is that this is a rapidly evolving technology, and there's these little machines being developed now that you could actually do this in the field. And I put this picture of Andreas here in Switzerland. He's actually diagnosing fire blight in this case, but he's running this assay in the orchard. And as this technology goes and these things become, they're a little bit expensive yet, but as it gets a little cheaper, we think that it'll be easier then to monitor fire blight just like we do for insects in the orchard. All right. So we've been using LAMP to look at this question of what does copper do as a delayed dormant for fire blight control. And so here's our walks again, and we've been with Rachel Elkins down here. She's down here. This is Rachel, and she's really led this project. Chuck Ingalls, who's here too, is involved now too, and we've been out walking orchards and looking at orchards where we just sprayed dormant oil on, or if we sprayed dormant oil plus copper, and we're getting results that look like this right here. So this is this was 2010 and 2011, where we just sprayed oil alone. We get detection assays that look like this. So we got mid bloom, full bloom, getting into petal fall, mid bloom, full bloom, petal fall, and you can see that the sanitation effect of the copper treatment is pushing our detection out pretty late in these two seasons. This is on average now, and see here. So then we did this again this year in 2012, and of course in 2012 it didn't work. We got exactly the same curves for both, whether we applied copper or if we didn't. And we think that was because we applied the copper way back in March, and then we had like a month of cold, wet weather, and the buds just sat there, and we think all the copper washed away. And actually, pears in California didn't bloom to almost May this year, and then we didn't see any kind of sanitation effect that we could measure. So this is an old technique, putting on copper for fire blight suppression before bloom, and you don't want to put it on during bloom because it roughs up the skin of the fruit. So this called delayed dormant copper is a sanitation. You can see the sanitation effect here from putting it on then. And in doing these LAMP surveys, we found that, at mid bloom, the overall probability of detection has been about less than 5% of the pathogen. But by the time we get to petal fall, we are finding the pathogen in about 50% of the sample. So you can sort of see that here. I mean, we're finding it in all the non-copper treated samples essentially in 2010 and about 60, but on average it's been about 50%. But we're getting a lot better picture about when the pathogen is becoming active and knowledge of that, and because of this we've been pushing harder to people to extend their control programs out to petal fall just because of the way these curves are shaped like this. Okay. I got to go fast. This is a systems approach. I got a bunch of things to talk about in 15 minutes, right? And so Rachel's been going in and getting the fruit finish data too, and I got to skip this here, but we haven't been able to detect a difference between where we use copper and where we haven't in fruit finish. So people are liking this approach. So quickly here, summary of LAMP survey. When is the pathogen active in orchards? Well, it depends on the orchard. We have some where we find it early, but late is more the norm. And I think we've educated people a lot about about fire blight and the pathogen activity, so they're not running out there right away at 50% bloom and putting their protection on because they're wasting their money. And we've changed some control programs like that. Does delayed dormant copper affect pathogen activity? Yes, it does. It seems to delay when the pathogen is detectable, which is a good thing, and allows more time, especially when we start talking about biological products to work out there and develop to the point where they are protective. And then I always get asked questions about, can I get lambsconning done in my yard? And I say that the technology is evolving rapidly, so perhaps soon that we can get this on a kind of a local level and people can do it for themselves. All right, so think about questions about lamp and delayed dormant copper for the panel. This next thing, I'm going to go pretty fast on it. It's probably been our biggest impact thing we've done in this project so far, is looking at the effects of lime sulfur on fire blight control. And as many of you know, as used for bloom thinning, it's used for bloom thinning in apples, and we wanted to know, does it provide a benefit to fire blight suppression, or stated another way, is lime sulfur a bactericide? Everybody knows it's a fungicide, but it really doesn't have any literature that says it's a bactericide. So if so, if it is a bactericide, if so, does it replace other fire blight control products in early bloom? And so the typical protocol for using lime sulfur for bloom thinning is to spray right after king bloom are open, which is about 20-25% bloom, and then repeat that again in 3 days. So you're looking at 70-80% bloom. So you let the king bloom come open on apples. We're talking about apples now. Go out there, hit them with lime sulfur, and then repeat that in about 3 days later for a second treatment. And then Harold will tell you that there possibly another treatment on the top half of the tree another 3 days after that. Okay. A lot of data here. So this would be just replicated orchard trials, just looking at lime sulfur and fish oil, in this case, lime sulfur and fish oil. This would be a standard here. This would be Blight band, then oxytetracycline. So this would be strikes per tree here. So you're going out here 150 strikes per tree, 200 strikes per tree, 200 strikes per tree, 80 strikes per tree, and this would be the water control as the blue bar. So you can see that if we just put lime sulfur and fish oil on 2 times, at the 2 times I just said, you control fire blight approximately 50% or so, 50, maybe 40%, 40 to 50% control of fire blight just from the lime sulfur. And we think that this effect is just largely due to fewer flowers out there, because we burned these flowers out now with lime sulfur. So in the way we do these experiments is we put the pathogen on after the second lime sulfur and fish oil. So the pathogen itself doesn't actually get hit with lime sulfur. So we put the lime sulfur on after we've done the thinning treatments, and sure enough, we get less fire blight, and we think that that's due to fewer flowers. But this year we did it sort of the opposite way, and we actually put the pathogen on flowers at about 70% bloom, and then came back the next day and hit them directly with lime sulfur. And so this would be the untreated control. We go back and sample the flowers and see how many bacteria actually on the flowers. So we're getting 100,000 bacteria on the flowers where we didn't do anything. Here's lime sulfur and fish oil. Now we've cut it almost 2 log units. So we're down here in the 10,000 cells per flower. And by the time we get out to the real high levels of lime sulfur, which are used, in really heavy bloom situations, we're down here, we've cut it almost 3 log units at this point. And so, and we go back this on these golden delicious that we had here, we got probably 50, 60 strikes per, we just were doing branches here, but 50, 60 strikes on the branches that we inoculated. You get down to this level and you don't hardly get any strikes at all, So the lime sulfur going on top of the pathogen on the flowers is knocking them down and definitely reducing fire blight risk out there. All right, so think about questions about bloom thinning. That's bloom thinning all in one slide, so it goes pretty fast. Last thing is integrated control, and can we achieve non-antibiotic control with the products that we have? And what we've done here is we've sort of split up fire blight control into 2 processes in the flower. And so the stigmas here are where the pathogen grows. That's where that epiphytic cycle goes around, and you get these big numbers of cells. But infection actually occurs down here in the floral cup. So the floral cup, there's these little openings called nectar thodes where the nectar comes out. Well, that's actually where infection occurs. So the bacteria grow up here, and then it slides down and goes in these nectaries. And you need to get a pretty big number up here before you get enough that actually can go in down here. That's how the disease cycle works. And so we've used this approach where we put on bloom time biological, which is a Pentoia glomerans strand, to colonize this. It's a gram-negative bacterial antagonist that colonizes this. And then we come in with oxytetracycline late in bloom, and that pretty much fills this floral cup. And we found that by doing that, we get better control than we get with 2 applications of that alone or 2 applications of oxytetracycline alone. So we've called this kind of an antibiotic integrated approach. And so we've tried to do the same thing with non-antibiotic materials. So if we go with bloom time biological, then Serenade Max as a substitute for oxytetracycline, or if we go for lime sulfur and fish oil to burn out stigmas and then put Blossom Protect on as something that we know can colonize the nectary, then we get another type of control program pattern on this same idea of integrated control. So here's one slide just showing this. This would be either Blight ban, which C9-1, which is a Pentoia glomerans, or bloom time biological, which is another Pentoia glomerans, put on at early bloom, and then they're followed by either oxytetracycline as the control, or with Serenade, which is a bacillus, but really you're just getting those antimicrobial lipopeptides in the fermentation of the bacillus, and that's how Serenade really works. And so same slide here, water controls, they're up to 450 strikes per tree, 200 strikes per tree, 200, so really heavy disease. So here's the Blight ban, then Oxytet, that works pretty good. Here is Blight ban once, then Serenade once, that's that gold bar. It works sort of so-so. If we double down on the frequency of materials, though, and go with, say, bloom time or Blight ban twice, then Serenade twice, well, then we're starting to we're getting better control, you know. So we've been recommending this for what was called the International Program for Pears. It still makes people nervous that this level of control isn't quite sufficient to be out there, but people are using this, and the important thing here is that the integrated idea seems to work, and that we're increasing the frequency of treatment, which increases the expense of the program somewhat. All right. Same thing here now, same idea, but now we're looking at this lime sulfur and fish oil, then Blossom Protect. And you can see here some pretty amazing data. I've got the streptomycin on here now. Streptomycin is this green bar, bloom time, then Oxytet. These are control standards. Now lime sulfur and fish oil, then Blossom Protect. We heard about Blossom Protect from one of the European talks this morning. It's a Ariobasidium pullians. It's a yeast-like organism. Pretty outstanding control of fire blight with that. Here's just the lime sulfur and fish oil alone again. So after the lime sulfur and fish oil goes on, then we're coming in with this Blossom Protect program after that, 2 applications, and you can see, you can see the additional benefits that we're getting. We're getting levels of control that are essentially on par with streptomycin in this case. All right. Couple things here. This slide doesn't come out real good on the screen here. This is a nectary of Pear that we took pictures of with a scanning electron microscope, and it's all those little bumps on there are yeast cells. So those yeast cells, and I took several of these. Just grab a flower that got sprayed with Blossom Protect, and that yeast is just all over these nectaries. What I'm trying to show here is this is stigmas right here. Am I on it? Do I got time yet? No? Okay. So I've got stigmas here, and they're colonized by the yeast, and they're colonized by the bloom time. We look at the nectaries. I've got yeast on the nectaries, but I don't have any of the bloom time on the nectaries. So I'm just about done here. All right. So can we achieve integrated control? Yes, using delayed, or can we achieve non-antibiotic control? Yes, utilizing delayed dormant copper sanitation in apples, lime sulfur to further delay pathogen buildup in flowers. In Pear, bacterial stigma colonizers to further delay pathogen buildup in flowers, and then utilizing Blossom Protect or Serenade Max at full and late bloom to protect that floral club. And compared to antibiotics, treatments are increased. And so questions here on all those things. And then we have a lot of sponsors, just like everybody else that has sponsored this research. And I'll stop.